Yeah, I have the right one.
And thanks y'all for sticking around for this part of the talk. I need to acknowledge the folks that have helped me with this research, particularly my advisor, Karen Mock, Justin DeRose with his dendrochronology wizardry, and Rick Lindroth, who's the sort of trade-off resistance tolerance that helped me out quite a bit. I know you guys have seen a lot of slides about the importance of Aspen, but I couldn't resist my short list. So Aspen does create quite a bit of habitat out west. It's often valued for forage and browse by livestock producers and wildlife folks. And although this is a picture that Bob had shown earlier down in the fish lake, it's not a fire-resistant species. It's often seen as a fire break. And on the left here, this is an actual BLM operation where they're removing the conifer component in a wildland-urban interface, trying to promote aspen growth to resist the area. And I don't think I need to stress the importance of how culturally and aesthetically important aspen is to it. So if it's not obvious yet, with the last two talks, the aspen paradigm is changing. We are experiencing a lot more clonal diversity in these landscapes than we once thought. These large clones tend to be triploid, and this is a recent discovery, we're actually making quite a stink about this. We do think that there's going to be some important ecological implications, especially in regards to management. And clonality of aspen is part of its strategy. These individual stems were often considered to be very weakly defended or ephemeral, where these clones persist in the landscape for quite some time. And those individual clones can respond quite uniquely from the one right next door experiencing similar environmental conditions. So aspen stems are susceptible to, this is Cytospora on the left here, probably the most common aspen canker that we see in the western United States. This is galleries of aspen bark beetle that we also see quite a bit. Aspen are host to hundreds of insects, and 9 of which of them are outbreak folivores. These are just 2 examples of them, and they're capable of creating quite a bit of canopy damage within one growing season and actually disrupting aspen growth for that season. And although I said stems of aspen are weakly defended, that's not to say that they're not defenseless. To say that they are defenseless. They create these phenolics, phenolic glycosides like salicortin and tremulacin. Here's the chemical formula if anybody's into that sort of thing. And the phenolic condensed tannins have also been linked to as an anti-herbivory compound. So trees have a decision to make. There's this common pathway where starches move along, and it gets to a certain point. It's like, am I going to grow or am I going to defend myself? And there's benefits of doing both, but you can't use the same starch to do both. It's to the dilemma of plants. And this is a pretty seminal publication by Herms and Matson, where they do a really good review of the literature up to that point, exploring all the various hypotheses and theories about plant defense and growth. To our research question, in aspen, are they displaying this pattern of a trade-off between growth and defense chemistry? And if there is there a clone effect? Because there's different clones in the landscape, is this one clone doing it more than that one? And, of course, is there a ploidy effect? If these clones are growing more as triploids, you would expect maybe that clone would defend itself less. So I'll get into the actual how we've done the study, the specifics here. This took place at Swans Flat, this very same place where Karen, if you were here for her talk, carried out a portion of her study. The elevation is about 2,400 meters, and the forest type is both aspen, pure aspen, and mixed aspen conifer. Okay, so this is a familiar map. Karen showed this, but it was in color. This is a blow up of this area. These individual shapes represent the clones, and black indicates a triploid organism, and white indicates a diploid. So I went into these areas where there are these, so these are all these squares, and I wanted to compare this clone to that clone. So I set up a plot, and each of those 50-meter points indicated by these guys, and in the middle there, I just randomly sampled aspen that were about 10 centimeters in diameter. And at each of those ramets or stems, I collected a representative canopy of what was going on in the leaves, and I also grabbed increment cores to measure growth. And I did this a total of 18 times across the entire landscape at Swan Flat, and I split it in half. I had 9 diploids and 9 triploid organisms, and that gave us a total of about 100 stems. So in the lab, we took one of those leaf samples from each of the trees, and using microsatellite analysis, as Karen mentioned earlier, we confirmed the clonal identity of all those stems within that one plot. And anything that was not part of that clone that we thought, we just discarded from the study altogether. The phytochemical analysis happened at Rick Lindroth's lab at the University of Wisconsin, and we looked at the percent dry weight of those phenolics that I mentioned earlier, condensed tannins. And I took the sum of the salicortin intermolation because they're highly collinear, and they're both functioning in this system. So that's how we looked at the defenses. And growth was determined by the actual growth rings of the last 10 years. But I wanted to, since we're only looking at the phytochemistry of this 1 year, we didn't want to compare growth since this tree originated. So we just kind of averaged the last 10 years. And the reason why I didn't use the last year is that last 10 years kind of quieted down the stochasticity of growth. So in order to account for this sort of like structured field design where you have ploidy up here and then clones underneath ploidy and then these individual ramets, nested frequency model is necessary. And we used SAS version 9.2 and the Glimmix procedure to create this linear model where growth was regressed by these various predictor variables, ploidy, genotype, phenolic glycosides, tannins, and age ended up being a very important variable in the model. Although we weren't looking at age specifically, it was necessary to leave it in there because of how significant it was in the relationship here. So here's the results. Does a trade-off exist between growth and defense chemistry? And if you're looking at those phenolic glycosides, salicortin and tremulation, there is a trade-off. If you're looking at all of the samples sort of pulled together, there was not evidence for a ploidy effect. So we were hoping that, or maybe thinking that you would see triploids, since they're growing more, to display this pattern differently than diploids, and that was not the case with this study. And there's a similar table like this that has a list of all 18 genotypes with really high p-values that I spared you from seeing, but they were there. So that gave us a little bit of heartburn. We were thinking all these clones are out there. They should be displaying this relationship, this trade-off. Since the trade-off does exist, at least with the phenolic glycosides, you would maybe see that with triploids, between triploids and clones. So we did some post hoc analyses. Okay, if there's these triploids and diploids, and I had this very split, very nicely designed study as far as the t-test would be concerned, I just compared the growth between the diploids and triploids. And did I see a difference in growth? The short answer is no. There was no difference. And you might be thinking, well, what did Justin find if you were here for his? Where he did see a difference. He was seeing that difference in growth at establishment, and it quieted down when it got down to when the trees got older. And I'm only looking at the last 10 years. So that would explain at least that portion of it. But the variances between the two were significantly different. So you did have more bigger organisms happening out here. Just the average wasn't kind of shifting the distribution over this way. So if people are seeing larger organisms and they're triploid, well, maybe they're seeing a big tree and they're kind of determining the ploidy level, and that's why. Because they're only kind of sampling those trees. This is speculative, but just trying to give some of those questions. So the difference between clones, if there's all these clones out there and there's a possibility of these clones doing very different things, why didn't that show up in the study? Is there even a difference between the phenolic glycosides between clones? And on the x-axis here, each one of these is an individual. These are all the 18 clones, and this is the percent dry weight. And it was highly significant between all of them. But the study wasn't replicated in the fact that I didn't have each one of these stands. I didn't have each genotype, so you can't really do a two-factor ANOVA. But a single-factor ANOVA was significantly different. And I wanted to see if this stand was maybe driving this relationship. So the best I could do for the purposes of this presentation was just, they're pooled together. So this is one stand of Aspen, and even in that one particular area, you can really see these differences jumping out between the clones, which we thought was cool. Did a similar thing for condensed tannins, same story. These areas, these stands did not seem to be driving this relationship. It really seems like it's clone. And I also did just a single factor ANOVA with growth, and it was significant. The p-value was, falling in barely below 0.05. Definitely not as much difference, but still there's a difference there, just not as much. So the take-home points: was there a trade-off? Yes, but clonal and plody effect were not there, but there was no evidence in the model showing that clones and plody were affecting it. Triploid stems did not grow faster than diploids, but that variance was different between the two. And individual Aspen clones differed in overall phenolics, and they differed in overall growth, but not nearly as much as those phenolics, indicating that maybe the landscape had a little bit more to do with growth landscape variation. And the implications that I'd like to draw on are similar to what Justin and even what Karen was saying. Was we're becoming aware that these landscapes are highly diverse as far as sexual regeneration and multiple clones in the landscape. They do have the tendency to respond differently. And I'm not saying we need to take a clonal-centric view of managing Aspen, but having that in the back of your mind when you're prescribing some silviculture treatment at Aspen is only going to help you down the road. And having this sort of genetic diversity in these stands is going to make these areas of Aspen quite a bit more resilient as well. So with that, I'd like to thank my committee and all of the folks that helped me along the way, especially the funding sources, the College of Natural Resources with the grant, and the Center for Integrated Biosystems at USU really helped out, and my most loyal field companion, my dog Ruby. And that's it.
Thank you.
Does anyone have any questions?
Yeah, there's time for a couple of questions here.
Yes.
Can Ruby smell the difference between the diploid?
I wish. Yeah, I'd get a lot more samples if she could.
So it's about your talk in terms of resistance or response, but where I'm coming from is that we've got some fenced off areas that are constantly regenerating. I mean, they're regenerating because the chicks are dying off, and you can pull them up really easily. Is it possible for the pathogens to get into the root system in the clones and sort of transfer that disease so that as you're getting suckering, you're also getting the disease kind of coming along with the suckering?
When you say you can